Hello, Dr. Fam.
All right, so participants, great. Keep completing the attendance form for us.
I can't see the attendance form. Where is it, please? Can you please?
Check, check the chat, the meeting chat. It's there.
Dr. Famakide, we can see your slides. Yes.
Thank you, sir. If you can put it in slideshow mode.
Dr. Thelma, you can read his citation to introduce him while he's finalizing his setup.
All right, please check your WhatsApp, Madam Ijoma. All right. We'll take the brief resume of Dr. A.A. Famikinde, FPSN, FPC Farm, FIIM, FADHRI, FSIS, FIPMA. I don't know the meanings of all the credentials. So Dr. Famikinde is a senior lecturer. He's the head clinical pharmacy and pharmacy administration, Faculty of Pharmaceutical Sciences, Achievers University, Owo, and resource person in oncology specialty, residency preceptor, project supervisor, and faculty board member of the West African Postgraduate College of Pharmacists, Lagos, Nigeria. He's a clinical pharmacist, pioneer oncology pharmacy specialist at the Federal Medical Center, Owo. He is a public health practitioner, an astute researcher by learning and practice, a trained administrator, information and communication technology specialist, an educationist, a bioethicist. He was a former staff of a tertiary health institution in Nigeria, Federal Medical Center, now a teaching hospital affiliated to Achievers University, Owo. The institution is noted for high professional and ethical practice standard, well organized and highly committed to make positive impact, field changes in the healthcare delivery system in Nigeria, and rose to the position of director, head of pharmaceutical services. He is a perfect combination of humility, intelligence, and quietness, HIQ. He is a focused goal-getter and a very consistent person. He is a fellow of the West African Postgraduate College of Pharmacists, fellow of the Pharmaceutical Society of Nigeria, fellow of the Institute of Information Management Africa, fellow of the Institute of Professional Managers and Administrator, fellow of Healthcare Informatics Society, and fellow of African Digital Health Research Institute. He is a member of many national and international academic and professional organizations. He has won many awards nationally and internationally. He has many postgraduate degrees that cut across health sciences, information, communication, technology, social sciences, and medical humanities. His unrivaled academic achievements, skills, and expertise, coupled with his humility, intelligence, diligence, honesty, God-given talent, and innovative capabilities, endeared him into the heart of whoever comes into contact with him at his various fields of endeavor. Participants, ESCO, distinguished ladies and gentlemen, please let's welcome Dr. Famekinde as our first facilitator for today's program. Thank you, Doc, and over to you. Dr. Famekinde. Hello, sir.
Okay, so that's yes. We don't have to do that first.
Hello, Doc. Can you hear us?
Hello, Dr. Famakide. We can see your slides. Can you please speak? Let's hear your voice.
So what about the co-presenters, Fikayo Adesanya and Amal Bongbasi? If they're on the call, maybe they can start off.
He's actually unmuted, so I don't really know what the problem is.
Maybe if someone can reach out to him.
Maybe he's
To
having.
him.
Okay, let me
Let
try and
someone
reach
reach
out
out
to
to
him.
him.
Please,
Yes.
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 Hello.
Hello, sir. We can hear you now. Please go ahead, but we can't see your slides anymore.
Hello, everyone. Can you hear me?
Yes, we can
Yes,
hear you.
sir.
Yes, we can hear you.
Sir, we can hear
And we
you.
can see you. Just share your slides and continue, please.
Please, can you see my slide?
Yes, we can see your slide.
We
Yes,
can see your slide. Kindly
and
put
we
it
can hear
in
you.
slow mode so that it's
No.
much quicker.
Good morning, everyone.
Good
Good
morning,
morning,
sir.
sir.
I'm delighted to give the first presentation for Oncology Pharmacy Practitioners Association of Nigeria workshop in collaboration with West African Postgraduate College of Pharmacists. The first of its kind coming up today. We are three that are supposed to present, but I will be presenting on our behalf. The other co-presenters are supposed to be pharmacist Fikayo Adesanya and pharmacist Amebong Basi. I want to appreciate their contribution to this presentation. And I want to appreciate Open, Chairman and ESCO, as well as West African Postgraduate College of Pharmacists for giving me this privilege. My outlines are as follows, and the topic of today is cancer cells biology and genetics.
Sir, Dr.
And it has
Famikide, please, can you put it in
topics
slideshow
which are
mode?
hallmarks of cancer, dysregulated signaling, apoptosis, metastasis, molecular oncology, and tumor microenvironment.
Sir, please, can you put your slides in
The outlines
slideshow
are as
mode?
follows. Have the introduction to cell biology, hallmark of cancer, dysregulated cell signaling, apoptosis in cancer, metastasis, molecular oncology, tumor microenvironment, and recent advances from 2023 to 2025. And we have some abbreviations and references.
Dr. Famakinde, please, the moderator has been asking you to.
As an
Hello,
oncology pharmacist.
Please put
It's
this on
like
Hello, can you hear me,
slideshow.
please?
Please
we
put
can
your
hear you,
slides
but
on
not
Hello,
hearing you.
slideshow.
okay.
Put it in slide share mode, please. Put your slide in slide share mode. Yes, thank you. Thank you. Continue, sir.
Hello, can you hear me, please?
Yes,
We can
sir,
hear
we can
you.
hear you.
Yes, as an oncology pharmacist, our responsibility are to take care of our patients who are in need of our services, and also to conduct credible researches that deals with cancer. And for this, I believe all of us know what cancer is all about. But let us remind ourselves that cancer is a disease characterized by uncontrolled cell proliferation caused by a series of cellular or genetic alterations or translocations. And a lack of controlled growth, cell divisions that lead to formation of tumors and invasions of tissues in proximity tumor cells. And also, it is characterized by genomic instability, evasion of natural cell death, as well as the ability to spread to a distant site and establish itself in that particular area. So, and is as a result of some gene mutation, because we are dealing with cell biology this morning. And these mutations affect genes that regulate cell cycle progression. It also has effects on DNA repair, apostasis, cellular metabolism, interaction with surrounding tissues. And we must note that cancer is not caused by a single mutation. It results from multiple genetic alterations, which are accumulated over time and leading to clonal expansion of abnormal cells. And it behaves like evolving ecosystem. That is, cells complete, adapt, and survive under selective priors such as immune attack and therapy. Cancer as a multi-step process. Cancer progression involves initiation, promotion, and progression. Initiation in the sense that it deals with DNA mutation, which is an irreversible genetic mutation that occurs in a protogen and tumor suppressor gene. And the promotion deals with clonal expansion, that is, the mutated cells gain proliferative advantage and expands clonally. While the progression, which is genetic instability and invasiveness, this occurs over time, in which the cells mutations accumulate and acquire invasive tendency and leads to metastatic and therapy-resistant properties. This explains why cancer develops over a decade. Now, what do we mean by genomic instability, which is one of the key points of cancer multi-step process? Genomic instability is the hallmark enabling cancer evolution. It includes chromosomal instability, that is frequent gains through losses of all chromosomes. It also involves microsatellite instability, that is errors in short repeated DNA sequence due to mismatched repair defects. It also involves defective DNA repair mechanisms in which the DNA cannot be repaired, and this increases mutation frequency. We also have a telomere dysfunction. This leads to chromosomal breakage and rearrangement. In all this, if they are not present, cancer will not accumulate mutations fast enough to progress. So cancer progression is from mutation, signaling deregulation, apoptosis evasion, microenvironment adaptation, and metastasis. That is the sequence, cancer progression sequence. Now, hallmark of cancer. The hallmarks framework was proposed by Douglas Hammer and Robert A. Weinberg. It defines common biological capabilities acquired during tumor development and organizes cancer complexity in shared traits across tumor types. These hallmarks are not sequential steps, but capability that cancer cells acquire during evolution. And they help us to understand why very different cancers behave similarly at biological level. We have the core hallmarks, which are sustaining progressive signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis, and activating invasion and metastasis. And we have emerging hallmarks, which are deregulating cellular energetics, avoiding immune destruction, and the enabling traits, which is the genome stability and tumor promoting inflammation. Now, cell signaling, which is a major concept in cell biology related to cancer. Here, the normal cells require signals known as ligands to tell the cell when to start or stop growing. Signal transmits into cell through proteins found on the surfaces of cell membranes known as receptor, and each ligand binding to a specific receptor. The receptors typically consist of 3 parts or domains. One is extending in an extracellular matrix, also within the cell membrane, and also one extending intracellularly. We have dysregulated cell signaling. This involves broken, overactive, or deficient molecular pathway in which we have the MAPK and PI3K stroke AKT EGFR. Now let me break this one down. We have the MAPK, which is mitogen activated protein kinase. And we have the PI3K AKT. This is a phosphatidylinositol 3 kinase. Phosphatidylinositol 3 kinase. That is PI3AK, which is a pathway. And it's an intracellular signals, like I've mentioned the other time, that we have 3 domains, or the intracellular signal transduction pathway that promotes metabolism. That is PI3K AKT. It's also enhanced proliferation, self-survival growth, and angiogenesis in response to extracellular signals. Now, I mentioned that we have 3 parts or domains. One is extending into extracellular matrix. The other one is within the cell membrane. And the third is extending intracellularly. But PI3K AKT pathway, which is intracellular signal transduction pathway, promotes metabolism, proliferation, cell survival growth, and angiogenesis in response to extracellular signals. And this is mediated through serine and threonine phosphorylation of a range of downstream substrates. And they have high affinity for cell surface receptors for many polypeptide growth factors, cytokinins and hormones. And these receptors have three functional domains, which is an extracellular ligand binding domain, a transmembrane domain, and intracellular tyrosine kinase domain. And this part is to stimulate the cell growth and proliferation. So they disrupt normal cell communication, leading to disease like cancer. It also have a role to play in diabetes and neurodegeneration. It is triggered by genetic mutation or environmental factors. And this imbalance causes uncontrolled growth, resistance to therapy, and impaired tissue or impaired tissue repair. Cancer disrupts normal signals transduction. And it takes the following pathways. We have the RAS map. We have the RAS map pathway, which play a role in proliferation. RAS means rat sarcoma. That is one of the most common genetic alterations observed in human and experimentally induced rodent cancer. It's a family of genes that makes protein involved in all signaling pathway that control cell growth and cell death. So that's for RAS-MAP pathway. And we equally have a PI3K AKT, which I mentioned earlier on, which is a phosphatidylinositol 3 kinase. So they play a very important role in survival and metabolism. And we equally have a P53. P53, this gene, like RB gene, is a tumor suppressor gene. And its activity stops the formation of tumors. But if a person inherits only one functional copy of P53 gene from their parents, they are predisposed to cancer and usually develop several independent tumors in a variety of tissues in early adulthood. So that is why you will see cancer runs in families. because of the inheritance of this type of gene, P53. Yes, I've also mentioned this earlier on, which I've just talked about. RAS-MAP enhances proliferation, and this pathway transmits extracellular growth signals to the nucleus to regulate cell division. And the mechanism in cancer is that mutations in RAS, especially KRAS, in 90% of pancreatic cancers, and BRAF. CRAS is a casting rat sarcoma virus oncogene homologue. And the mutations are present in approximately 25% of tumors, making them one of the most common gene mutation linked to cancer. That is CRAS. 90% also involved in 90% of pancreatic cancer. Whereas BRAF, which is Morin sarcoma viral oncogene homologue, the homologue B, is a gene found in chromosome 7, that is 7q34. The protein also plays a role in cell growth by sending signals inside the cell promoting, among other functions, cell division. So that is BRAF. And the result of this is sustained constitutive activation, derives continuous proliferation, and inhibits apoptosis, that is cell death, and promotes angiogenesis. Earlier on, I talked about PI3K-AKT, which is also for survival and metabolism. This axis is a major controller of cell survival, growth, and metabolism in response to nutrients and growth factors. The mTOR, I've not talked about that. mTOR is mammalian target of rapamycin. It's a protein kinase that controls cellular metabolism, catabolism, immune responses, autophagy, survival, proliferation, and migration to maintain cellular homeostasis. So the mechanism in cancer is that they are often activated by gain of function. That is GOF. Gain of functions mutation. And we have this in PIK3CA gene, or loss of function in tumor suppression in PTEN. And this results in tumor cell survival, that is resistance to apoptosis, and they drive massive anabolic metabolism. And this is what we call Warburg effect. Warburg effect. And it fuels rapid growth via the mTOR. mTOR so The p53, which I've earlier discussed, is known as the guardian of the genome. p53 stops the cell cycle to to allow for DNA repair or triggers apoptosis, that is programmed cell death if damage is irreparable. The good news here is that this gene can be repaired through gene therapy or drugs that restore or inactivate p53. And the drug aims to fix mutated p53 genes and protein. So the mechanism in cancer here is TP53 is mutated or inactivated in over 50% of human cancers, allow cells with damaged DNA to continue dividing. But like I said, it can be repaired. If the DNA continue to divide, it results into evasion of apoptosis, genomic instability, uncontrolled cell survival despite severe genetic damage. Yes, apoptosis, death, cell death. Is a highly regulated form of cell death initiated either through external signals, that is the extrinsic pathway, or internal stress, which is the intrinsic mitochondrial pathway. And the pathway leads to the inactivation of phasphases, cysteine, proteases that play a crucial role in executing programmed cell death. In an average human adult, 50 to 70 billion cells undergo the process of apoptosis daily. And broadly, three main mechanisms of biochemical changes can be observed in apoptosis. And one is activation of phasphases, DNA and protein breakdown, while the third is membrane changes and recognition by phagocytic cells. Like I mentioned earlier on, we have a phosphatidylserine. A phospholipid flips out from the inner layers of the cell membrane and become expressed on the outer layer of the cells. And this flipped expression allows for early recognition of apoptotic cells by macrophages, which results in engulfment and phagocytosis of the cell without the release of cellular components. And these are all that I've been discussing about concerning a programmed cell death. We have the normal cell, then there'll be cell shrinkage, chromatin condensation, then going to membrane bleeding, and the next stage is nuclear collapse containing bleeding. Then we have a apoptotic body formation, which later leads to lysis of apoptotic bodies. We equally have this. That's the mitochondria. We have cytochrome C, the adaptor protein, that is a receptor ligand interactions. We have the FAS, and we have initiators phasphases that I mentioned earlier on, leading to executional phasphases which can either lead to breakdown of cytoskeleton, or endonuclease activation. When it leads to endonuclease activation, it finally leads to DNA fragmentation. And this further lead to cytoplasmic bleb. And we have an apoptotic body, the ligands for phagocytic cell receptors, and taken over by the phagocytes. These are the key strategies to resist apoptosis, because this is very important in therapy. These strategies primarily focus on inhibiting the intrinsic mitochondrial pathway, the extrinsic death receptor pathway, or both. And we have the upregulation of anti-apoptotic BCL2 family proteins. And BCL2 is a BCL leukemia lymphoma 2 protein. The cancer cells frequently overexpress protein as BL2, BCL2, BCL, XL, and MCL1, which binds to and inhibits the pro-apoptotic protein BAX and BAC, all leading to the prevention of mitochondrial outer membrane permeabilization. Mitochondrial outer membrane permeabilization, MOMP. And we equally have down regulation, which is the loss of pro-apoptotic proteins. In this case, the cancer cells often lose or reduce the expression of BAX, which is BCL2-like protein 4, as well as BAC or BH3 only, and which all these are necessary for the initiation of apoptosis. We equally have overexpression of inhibitor of apoptosis protein. And the proteins such as XIAP and IAP, I2, 1 and 2, or surviving are frequently elevated. And after this, directly binding to and inhibiting the capsaicins, which I mentioned earlier on, and the executioner. The executioner, which is here, will be inhibited because it normally leads to either the endonuclease activation that leads to DNA fragmentation or the breakdown of cytoskeleton. Then we have inactivation of P3 tumor suppressor. The mutation or deletion of TP53 gene occur in about 50% of human cancers, while disabling this P3 mediated induction of pro-apoptotic genes will come up in response to DNA damage. I mentioned earlier on that this can be repaired using a gene therapy or other drugs. can work on the repair. Now, let's come to this regulation of death receptor signaling, the extrinsic pathway. We have the C-flip overexpression, and this increases in the cellular phase inhibitory protein, which later inhibits the activation of caspase 8. And the co-receptor expression, this is overexpression of receptor that bind to death ligands like trail, but lack functional death domain. Sorry, please. Then the decreased death receptor expression. This reduces surface expression of death receptor using the DR4 or DR5. When we talk about DR4 or DR5, this is death receptor 4 and death receptor 5. And the C-flip is cellular flight-like inhibitory protein. Cellular flight-like inhibitory protein. And the activation of survival signaling pathway, this one leads to hyperactivation of PI3K, AKT, which I've earlier mentioned, as well as RAS-RAF, MEK, ERK, MEK, ERK. And all this pathway promotes survival and stabilizes the anti-apoptotic proteins like MCL1. We equally have the Warburg effect, which I mentioned earlier on, which is metabolic alterations. This increases glycolysis and reliance on the pentose phosphate pathway. And these cell cancer cells generate energy and avoid apoptosis. And they are often mediated by AKT-dependent hexokinase binding in the mitochondria. And this prevents cytochrome C release. Earlier in the diagram, we saw the bubbling, membrane bubbling. And the membrane bubbling will lead to nuclear collapse, and the bubbling continue until apoptotic body formation, which ultimately leads to lysis of apoptotic body. So in this case, bubbling shield formation will stop apoptosis. So cancer stem cells may be used, and it may use an emergency program where apoptotic bodies, blebs, fuse to form Veblen shield and allow the cell to bypass apoptosis and survive. Now, metastasis. How does this one occur? Metastasis is a multi-step process encompassing 1, local infiltration of tumor cells in the adjacent tissue. It also involves transendothelial migration of cancer cells into vessels known as intravasation. And 3, it leads to survival in the circulatory system. As well as extravasation and subsequent proliferation in competent organs. Those are the process of metastasis. It's a multi-step. Local infiltration, then transendothelial migration of cancer cells into vessels known as intravasation, survival in the circulatory system, then extravasation and subsequent proliferation in competent organ. So let's take them one after the other. We have the local invasion. Cancer cells detached from the primary tumor invade the surrounding extracellular matrix and penetrate local tissue, and which often using matrix degrading enzymes, the MMP. Then we have intravasation. The invasive tumor cells enter the blood or lymphatic vessels, and this process can be aided by tumor-associated macrophages or by collective invading cells. Then we have circulation survivor. Without surviving in the system, circulatory system, it cannot progress. So it has to survive there. And once in the bloodstream, the cell becomes circulating tumor cells. And they must survive shear stress and cell death due to lack of adhesion, as well as immune system clearance. Without all this process, tumor cells cannot survive in the system. Then we have extravasation. The surviving circulating tumor cells leave the vasculature by adhering to the endothelial cells of capillaries in distant tissues and migrating through the vessel wall into the parenchyma. And we have secondary colonization. The excavated cells adapt to the new microenvironment. We are going to talk on the microenvironment later during the course of this lecture, and how it plays a prominent role in cancer progression. So we have the adaptation in the new environment. Then they have to survive and begin to proliferate, forming micro and eventually macro metastasis. The steps: local invasion, intravasation, circulation survival, extravasation, and secondary colonization. The steps in metastatic cascade. Now, molecular oncology. This deals with the study of genetics, epigenetic and molecular changes driving cancer development, focusing on how these alterations affect cellular behavior. It utilizes technologies like genomics, transcriptomics, and liquid biopsies to guide personal treatments, identify novel therapy targets, and monitor disease progression. And key aspect of molecular oncology includes the mechanisms, the techniques, clinical applications, and diagnostics. Mechanisms, this investigates how oncogenes and tumor suppressor genes drive uncontrolled cell proliferation. And also the techniques employs genomic sequencing, tumor imaging, and functional models to analyze tumor biology. While the clinical applications enable precision medicine by tailoring therapy, tyrosine kinase inhibitors to a patient's specific molecular profile. And it's also used in diagnostics. That is, uses liquid biopsies to analyze circulating tumor DNA from blood samples. We have molecular oncology therapies, immunotherapy, which are targeted approach to cancer therapy. All these you get to know in the subsequent presentations during the course of this workshop. A targeted approach to cancer therapy where actual immune cells of the patients and the genes are manipulated to produce an anti-tumor response. We equally have chimeric antigen receptor T cells immunotherapy, as well as gene therapy. Oncogenes. An oncogene is a gene which in certain circumstances can perform a cell into a tumor cells. It's a mutated form of normal gene called a proto-oncogene that manages cell growth when proto-oncogenes mutate into oncogenes, and they cause cells to grow and divide uncontrollably, which eventually the abnormal cells may form tumors. The proto-oncogene can become an oncogene by a relatively small modification of its original function. And there are three basic methods of activation. We have a mutation within a proto-oncogene, which causes a change in protein structure or causing an increase in protein activity or an increase in the amount of certain protein, resulting in multiple copies of gene, leading to overproduction caused by an increase in gene expression through mutation in the promoter region, or through an increase of protein stability, prolonging its existence, and thus its activity in the cells. And thirdly, is the gene duplication. That is one type of chromosomes abnormality. So, increase in amount of protein can result, and all this can result in increase in gene expression, increase in protein stability, and gene duplication. Then a chromosomal translocation, another type of chromosome abnormality, This is where rearrangement of DNA that causes gene overactivity or fusion of protein occurs. And there are two different types of chromosomal translocation that can occur. One is translocation events, which relocates a proto-oncogene to a new chromosomal site that leads to higher expression. Translocation events that relocates a proto-oncogene to a new chromosomal site that leads to higher expression. And two, translocation events that leads to a fusion between a proto-oncogene and another gene that creates a fusion protein with increased cancerous to oncogenic activity. And these are the diagram to show this. You have the proto-oncogene. You see that you have a chromosome rearrangement. You have a nearby regulatory sequence cause normal protein to be overexpressed, or fusion to actively transcribe gene overexpresses fusion protein, or fusion protein is hyperactive. Then you can equally have deletion or point mutation in the coding sequence, DNA to RNA. A hyperactive protein is produced in normal amount. And in gene amplification, you have a normal protein is overexpressed. So oncogenes, we have the examples, we have the RAS. And we have the NYC, we have the BCR, HER2, stroke new, and also EGFR. Let me just talk about this. RAS often mutated in pancreatic or in the pancreas, lungs, and colon cancers. While the MYC, is frequently associated with leukemia and lymphoma. The NYC signaling it functions as a transcriptional signal amplifier, and it accumulates in the promoter or enhancer region of all active genes and cause transcriptional signal amplification. So that's why how it works, and it's associated with leukemia and lymphoma. The difference between RAS and MYC is that MYC is a family of transcription factors, C1 and NYC, whereas RAS is a family of guanosine triphosphates, that's GT-pases. And we have the H, we have the K, we have the NRAS. So MYC generally localizes in the nucleus, while the RAS is localized in the cytoplasm. That's the difference between those two. Then the key factors in chronic myeloid leukemia, that's a BCR, and ABL1. And we have the HER2, which is common in certain breast cancer, and EGFR, which is implicated in the lung adenocarcinoma. Let's quickly look at this chart. The BCL, like we have mentioned, is just like a summary of what we have discussed. BCL2 is anti-apoptosis, common in lymphomas. Then we have BCR-ABL fusion gene, non-receptor tyrosine kinase. It's also common in chronic myelogenous leukemia. And the HER2 NEU, which I mentioned the other time, is a epithelial growth factor family receptor tyrosine kinase.
It's up to 50.
It's common also in breast cancer and Schwann cells. We talked about the NYC. I mentioned is a transcription factor common in neuroblastoma, leukemia, and lymphoma, which we have mentioned. We talked about RAF, serine, tyrosine kinase, which are common in liver and lung cancers, and so on and so forth. Then let's talk about tumor suppressor gene. A tumor suppressor gene or anti-oncogene is a gene that regulates cells during cell division and replication. Why a tumor suppressor gene is mutated, it results in a loss or reduction in its function. And in combination with other genetic mutations, this will allow the cell to grow abnormally. And the loss of function for these genes may even be more significant in the development of human cancers compared to activation of oncogenes. The TSGs can be grouped into the following categories. We have the caretaker gene, we have the gatekeeper genes, and more recently, landscaper genes. That is tumor suppressor genes. The caretaker genes ensure stability of the genome via DNA repair, and subsequently, when mutated, allow mutations to accumulate. Whereas the gatekeeper genes directly regulate the cell growth by either inhibiting cell cycle progression or inducing apoptosis. While the recent ones, the landscapers genes, regulate the growth by contributing to the surrounding environment when mutated and can cause an environment that promotes unregulated proliferation. And in most cases, tumor suppressor protein inhibits the same cell's regulatory pathway that are stimulated by the products of oncogenes. While the tumor suppressor gene have the same main function that have various mechanisms of action that their transcribed products perform, which include the following: intracellular protein that controls gene expression of a specific stage of cell cycle. And by the time we'll be talking about the chemotherapy, you know that some of the things that we are talking here will be referred to in the choice of the chemotherapy that you are going to apply on our patients. If these genes are not expressed, the cell cycle does not continue and effectively inhibiting cell division. We have the PRB and PI6. The transforming growth factor, TGF beta, and the adenomatous polypsy coli, the APC. We equally have the checkpoint control protein that triggers cell cycle arrest in response to DNA damage or chromosomal defects, especially in breast cancer type 1 susceptible protein. And we equally have protein that induces apoptosis. If damage cannot be repaired, like I mentioned when I talked about the P53, that it can be repaired, but if it cannot be repaired, then the cell initiates programmed cell death to remove the threats it poses to the organisms as a whole. We equally have cell adhesion. Some proteins involved in cell adhesion prevent tumor cells from dispersing. They block the loss of contact inhibition and inhibit metastasis. Also, these proteins are known as a metastasis suppressor. We have a protein involved in repairing mistakes in DNA. Here, the caretaker genes encode proteins that function in repairing mutation in the genome, preventing cells from replicating with mutation. Also, we have increased mutation rates for decreased DNA repair that leads to increase in activation of other tumor suppressors and activation of oncogenes. The P53 and DNA mismatch repair protein 2. Also, we have certain genes that can also act as tumor suppressor and oncogenes. The proto-oncogenes with tumor suppressors function. These genes act as double agents that both positively and negatively regulate transcription. We have the Notch receptor, we have the TP3, 53, and we have FAS. Under the proto-oncogenes and tumor suppressor genes, they play important roles in preventing the development of tumor. The proto-oncogenes supports healthy cell growth and division, while the tumor suppressor gene regulates cell division. We can see from the diagram here. Then the mutations in proto-oncogenes, converting them into oncogenes, and mutations in tumor suppressor genes can lead to uncontrolled cell proliferation and cancer development. The balance between the two types of genes is crucial for cellular homeostasis, and disruption is a hallmark of cancer progression. Understanding these mechanisms allows for targeted therapy, such as using drugs to counteract the effects of activated oncogenes or attempting to restore the function of lost tumor suppressors. So this we talked about in the immunotherapy and other cancer therapy. Now let's quickly talk about the tumor microenvironment. which is very, very important in metastasis. The tumor microenvironments consist of tumor cells, tumor stroma cells, including stroma fibroblasts, endothelial cells, and immune cells like microglia, macrophages, lymphocytes, and the non-cellular components of extracellular matrix such as collagens, fibronectin, aronin, laminin, and among others. As the heart of tumor microenvironments, tumor cells controls the function of cellular and non-cellular components through complex signaling network to use non-malignant cells to work for their own benefit. The tumor cells hijack different cellular and non-cellular non-malignant components of tumor microenvironments to promote their own growth and survival under hostile conditions. And the mediators such contacts can be soluble factors like chemokines, cytokines, or growth factors, or those that enable horizontal genetic structural material transfer, including cfDNA, apostotic bodies, and CTCs, as well as exosomes. The components of a tumor microenvironment. We have the cellular component. We have cancer-associated fibroblasts. This derived from the normal fibroblasts, mesenchymal stem cells, or epithelial cells. Via epithelial mesenchymal transition, they secrete growth factors such as a transforming growth factor, beta, also vascular endothelial growth factor, VEGF, which have come across earlier on. Also the FGF, fibroblast growth factor. They promote tumor cell proliferation, extracellular matrix remodeling, angiogenesis, and drug resistance. And we know that all these ones are very, very important for an invasive cells to stabilize and continue to proliferate in a new site. So cancer-associated fibroblasts create a fertile soil for tumor expansion. We also have tumor-associated macrophages, often polarized by M2-like phenotype. They secrete interleukin-10, which is a immunosuppressive, and also TGF-B, that is transforming growth factor beta, as well as a vascular endothelial growth factor for angiogenesis. They suppress cytotoxic T cells and promote invasion and metastasis and enhance angiogenesis. High tumor-associated macrophages density correlates with poor prognosis in many cancers. We also have T cells, cluster differentiation 8, that is CD8 T cells. The anti-tumor activity. The regulatory T cells suppress immune response, and tumor exploits immune checkpoints such as PD-1 and PD-L1, as well as cytotoxic T lymphocyte-associated protein 4. All these immune suppressions allows tumor immune escape. We have endothelial cells. They form tumor blood cells, blood vessels, responding to vascular endothelial growth factor signaling. And tumor vasculature is abnormal, leaky, and disorganized. All these promote metastasis and aposia. We equally have extracellular matrix remodeling. This provides structural support, but is heavily modified in cancer. The key changes include increased collagen deposition, cross-linking lysyl oxidase, increased matrix stiffness, and the consequences are enhanced tumor cell migration, as well as activation of integrin signaling. This also leads to activation of focal adhesion kinase. It promotes epithelial mesenchymal transition and stiff extracellular matrix mechanically activates oncogenic pathway. Now, these are the roles of oncogenes in the expression. We have the aposia, which we mentioned the other time. Rapid tumor growth causes oxygen deficiency. And when this occurs, the aposia stabilizes the aposia inducible factor 1 alpha. And the next sequence is that aposia inducing factor 1 alpha induces VEGF, which is angiogenesis. Also, glucose transporter 1, which increases glucose uptake, and also glycolytic enzymes, which leads to Warburg effect. And Warburg effect is an increase in the rate of glucose uptake and preferential production of lactates, even in the presence of oxygen. And this will promote angiogenesis. It increases invasiveness. It creates acidic microenvironment, as is the lactate accumulation. And the acidic pH suppresses immune cells and enhances invasion. Let me mention a few things with this glucose transporter 1, which increases glucose uptake. It's facilitated glucose transporter member, and that one is SS2A1, which is a unipolar protein that in humans encoded is facilitated transport of glucose across plasma membranes of mammalian cells. And in this case, it's as a result of aposia inducible factor 1 alpha. So we have angiogenesis in the tumor microenvironment. In this case, tumor cannot grow beyond 1 to 2 millimeters without blood supply. So, and the key driver here is vascular endothelial growth factor. And this is triggered, which we mentioned here, is triggered by vascular endothelial growth factor, triggered by aposia, and oncogenic signaling, the RAS and MYC that we mentioned earlier on. And the effect of this is supply oxygen and nutrients. It provides route for metastasis, and it produces abnormal vessels in which drug delivery, we form a drug delivery barrier. Then the role of in the tumor progression, we have inflammation and cytokine signaling. Chronic inflammation promotes tumorigenesis. And the key cytokines here is IL-6, which is interleukin 6. This is an interleukin that acts as both a pro-inflammatory cytokine and an anti-inflammatory myokine. In human, it is encoded as IL-6. We equally have a TNF alpha, that is tumor necrosis factor alpha. This is a cytokine that has pleiotropic effect on the various cell types. It has been identified as a major regulator of inflammatory responses and is known to be involved in pathogenesis of some inflammatory and autoimmune diseases. And here also we have the TGF-beta. This is a transforming growth factor. It is a multifunctional cytokine expressed by almost every tissue and cell type, as earlier discussed. We equally on all this activates NF-kB pathway. NF-kB pathway is nuclear factor kappa B pathway. And this is an ancient protein transcription factor and considered a regulatory of innate immunity. And it links pathogenetic signals and cellular danger signals, thus organizing cellular resistance to invading pathogen. And this results in an increased proliferation. We have STAT3 pathways too. STAT3 pathway, that is signal transducer and activator of transcription 3. Signal transducer and activator of transcription 3. It is a transcription factor as well in humans. It's a member of STAT protein family, and it plays an important role in normal development, particularly hematopoiesis, and regulates cancer metastasis by regulating the expression of genes that are crucial to cell survival, cell proliferation, invasion, angiogenesis, and tumor immune evasion. So the key cytokines, they activate these pathways, and the result is increased proliferation, reduced apoptosis, enhanced metastasis, and immune evasion. Now, immune evasion mechanism in a tumor microenvironment. Tumor suppresses immune destruction by PDL1 expression. And PDL1 is program death ligand, which we have mentioned earlier on. It inhibits the T cell activation. Then we have the recruiting treg and MDSC. Recruiting treg to tumor sites is regulated by chemokines produced in the tumor microenvironment. And we have a CCL22 ligands, CCR4 doing that. And CCL2 is also an antimicrobial protein with bactericidal activity, and it could be an immune marker in associated vasculitis. So we have a CCR7, which I mentioned the other time as a CC chemokine receptor type 7. So that's the ligand and also the CCR4. They interact with these ligands and play a significant role in cancer cell migration and metastasis formation. So we equally have, it will also secrete IL-10, TGF beta, and also metabolic competition, that is glucose depletion. So that's the tumor that tumor suppress immune destruction just by all these processes. It's either PDL1 expression occur, then recruiting treg, secreting IL-10 and TGF beta, as well as metabolic competition, that is glucose depletion. And the outcome is T cell becoming exhausted and immune tolerance develops. So the role in tumor progression as well. A tumor can change its microenvironment, and the microenvironment can affect how a tumor grows and spreads. This depends on cancer cell intrinsic factors, which we have already mentioned earlier on, the oncogenes, the tumor suppressor loss, then the genomic instability. And also, it includes the extrinsic factors, that is stroma support, immune suppression, angiogenesis, aposia, and ECM modeling. All this we have discussed earlier on. We've seen how they play a role in metastasis. Then the ECM modeling. The tumor behaves as an ecosystem rather than isolated malignant cells. The tumor microenvironment and metastasis. The tumor microenvironment facilitates each step of metastasis. All this also we have touched. The EMT induction via TGF beta, then ECM degradation via MMP, then intravasation via leaky vessels, survival in circulation aided by platelets, and a pre-metastatic niche formation. Now, the primary tumor will release exocymes, cytokines, growth factors, and these prepare distant organs for colonization. Then tumor microenvironment and therapy resistance. Tumor microenvironment contributes to resistance through hypoxia, radio resistance. Then we have the CAF. That is mediated drugs with sequestration, immune suppression, leading to immune therapy resistance, the ECM barrier, leading to reduced drug penetration. This is why targeting cancer cells alone is insufficient. Focus has shifted towards ecosystem-based therapy, not cell-centric therapy. And here we have therapies targeting the tumor microenvironment. We have anti-angiogenic drugs, VEGF inhibitor. We have immune checkpoints inhibitor, which we mentioned earlier on. We have CAP targeting therapies. We have TGF beta inhibitors. And we have CAR-T therapy modeling immune tumor microenvironment. For the CAR-T, that is the T-cell therapy. Cells from the body and genetic changes them so they can fight the cancer, and it is most often used to treat cancer cells such as leukemia, lymphoma, and multiple myeloma. So modern oncology increasingly targets both tumor cells and other microenvironments. We don't just target tumor cells alone. We also work to target the microenvironment. And you will see some of this when we are talking about the anti-cancer agents, different classes, and their mode of action. Yes, these are some of the abbreviations. And these are my references. Thank you very much for listening. I'm done. Thank you for your rapt attention. Hello.
Thank you, sir.
Hello.
Thank you, Dr.
Thank
Famatunde.
you, sir. Thank you, sir.
It's okay, thank you.
Thank you for that presentation.
Although this is the foundation, this is the foundation part of oncology. A lot of people run away from this because of cell biology. But as pharmacists, most especially as oncology pharmacists, we need to know this because it's the foundation of other areas that we touch in caring for our patients and also in formulating pharmacotherapeutic plan for the cancer patients. Thank you very much.
Thank you, sir. Thank you.
Thelma, are you on?
Very much.
Yes.
Yes, Prof, I am.
Okay.
I don't know whether we'll take questions or they should put them down when most of the presenters facilitators make their submissions or presentations, then we'll take the questions.
Okay, no, they finished the presentation. He presented for the whole team.
Yes.
Right. So then it
This
means
is time
we
for
can take
question and answer.
questions.
Yes.
Yes.
All right. So our dear participants, it's question and answer time. Kindly use the react button if you have any questions, and you'll be allowed to ask your question. We are at question and answer time. Thank you very much, Dr. Famakinde, for that presentation. Participants, kindly ask your questions. Use the react button. Just raise your hand and you would be given the opportunity to ask your question.
Okay, good afternoon, everyone. Can you hear me, please?
Yes, good afternoon.
Yes,
Please
you
go
can hear
ahead.
me. So,
Yes.
thanks to Dr. or Professor Famakinde for the presentation. It's not particularly directed at him, but to the team. I wanted to know if we're going to get the slides to study further after now. That's what I wanted to ask.
Yes, the slides will be made available. All the materials for this webinar will be made available to all participants.
Okay, thank
Dr.
you.
Okay. Dr. Effie Williams, please ask your question. Afterwards, 102, you can ask your question. Dr. Williams, kindly unmute and ask your question or make your submission.
Good morning, everyone.
Good
Want to
morning,
want to commend Dr. Famakinde for that beautiful presentation. Initially, for someone who is not exposed to oncology, pharmacy, the whole thing looks like Dr. Famakinde is speaking Greek. But the beauty of it is that with time, you find it very interesting. It gives the assurance that cancer is not a myth. It is something that we know is happening every day. And when you have that kind of understanding, you don't run away from it. To every problem, there's a solution. You face it and you're able to achieve. The summary of what we have learned so far is that the cell divides normally and it has a lifespan for that division. It's supposed to die after some time. But because of the mechanism of the cancer cells, to escape this cell death, it continues to grow and grow and grow, and it now causes problem for nearby cells and other cells that are far away. That is just a summary. So it's not a myth. It's something that we can fight. We can win the fight against cancer. Thank you so much.
Thank you very much, Dr. Williams. Serial number 102. Kindly unmute and make your submission.
Yeah, thank you, Professor Famakinde, for such a wonderful presentation. I just have two questions to ask, having listened to this basic cancer cell biology and also genetics. Number one is what is the role? What is the role of glucose in cancer growth? That's one. Number two, I am in natural product chemistry, and I needed to know if, though we're still yet to get there, what should be the target? What should be the target molecules in possibly handling the apoptosis of cancer cells, which will really aid my research in my area of oncology. Thank you so much.
Hello, Dr. Zia, we can't hear you again, or am I the only one? Hello, Dr. Thelma.
Yes, madam.
Can you hear him?
The participant or Dr. Famakide?
No,
I'm
the
here.
person that was asking question, that's Dr. Zia from UNN.
All
We
right.
can't hear him again.
I'm
Hello.
here.
Hello, can you hear me now?
Okay.
Yes, yes.
Yes, I just asked two questions. Number
Yes.
one is the place of increased glucose in the body system in cancer growth. I've been listening to the cancer cell biology and genetics. And number two, I said I am in natural product chemistry, and I've listened to the progressions, and I needed to know where target molecules could be engaged in the apoptosis of cancer cells. These are areas I needed clarification from Professor Famakinde, if he can help me out. Thank you so much. I don't know whether I was heard.
Yes, we heard you.
Yes, we heard you. We heard
you.
you.
Famakide, did you hear him, please?
It's for Famakinde to respond. That's if he's still there.
He's here, but he was complaining that he wasn't hearing us. He called me and was saying he wasn't hearing anything. Hello, sir. Dr. Famakide, can you hear us? There is a question for you. Let me try to get.
I can't hear from here. Hello.
Hello, Dr. Famakide, we can hear you. Dr.
Yes, I can't hear you.
Dr. Zia, please, can you put the question on chat box so he can read it there? I think he's having issues with his speaker. Hello, sir.
Okay. Let me do that.
Yes, sir. Thank you so much. While you're doing it, I just want to make a comment. Dr. Fomakide, please, if you can hear me, he's putting the question on chat box, so you can respond to his question, please. So I just want to appreciate Dr. Fomakide for this wonderful presentation. The truth of the matter is that cancer cell biology sometimes, just like Dr. Williams said, initially it will just be sounding like it's difficult for you to grab. You appreciate this topic more when you are in clinical practice, because you will begin to understand why some cancers respond well and why some don't respond well. I will give you a scenario. If you've managed clear cell renal cell carcinoma, is one of the cancers that don't respond to chemotherapy. So you're left with targeted therapies. The first patient we managed when I started practicing, when we were about to go for the multidisciplinary team, because in my center, for every patient, we do multidisciplinary team. So the oncology pharmacy team will have to review the patient and prepare their submission during the multidisciplinary team. So when we were doing our review, we discovered that renal cell carcinoma does not respond to chemotherapy, and over 80% of clear cell renal cell cancer is caused by mutation of the VHL gene. VHL gene is a tumor suppressor gene. And that is where the major problem is, because in the past, there was no drug that could target that mutation. But recently, there is a new drug, belzutifan, that targets that, but it's still in clinical trials. I think it has passed the phase 3 clinical trials. We hope that we'll have such drug in Nigeria. So, but what we do in managing renal cell carcinoma is we use targeted therapies and angiogenesis inhibitors. That's the multi-kinase inhibitors, the small molecules multi-kinase inhibitors, or the large molecules, the bevacizumab and all that. If you don't use them to manage the renal cell carcinoma, no matter what you do, the patient will just continue to deteriorate, and the next thing, the patient will just pass. But if you use those targeted drugs that target angiogenesis, because in kidney cancer, there are lots of blood vessels. So the only way you can help that patient is just to target the blood vessels, because what is driving the mutation is tumor suppressor gene, and there is no drug to target it. Chemotherapy doesn't help. So you begin to appreciate the knowledge of cancer cell biology when you are in clinical practice. It will begin to make sense, because you're seeing the disease condition, you're seeing the drug, you're talking about the multi-kinase inhibitors like lenvatinib, axitinib, and you know their mechanism of action, you know what they are used for. And there is a common say in cancer space, when a cancer is being driven by mutation in oncogenes, it's easier to treat than when it's being driven by mutation in tumor suppressor gene. So you begin to know the difference between the oncogenes and the tumor suppressor gene. So this topic is quite a very interesting topic for anybody that really wants to specialize in oncology pharmacy. So I want to encourage us, when you get the slides, sit down and read the slides. There is a course that we packaged as a boost for all the participants that are attending this workshop. We are going to make it available at the end of the training. It's an online course, but it's free. You can sit down and do the course. All these courses are there in detail, in addition to what you will get from our facilitators. You will sit down and do it at your own time, whenever you want. There is also a certificate attached to it. So we are going to make the link available. We did it Open did it in collaboration with BVGH. I don't know if you've heard about BVGH. BVGH is a multinational NGO that does a lot of training in oncology. So we'll make it available for the participants at the end of this pre-conference workshop. So when you go there, you also see materials and videos, lectures on cancer cell biology, so that you'll be able to understand more about it. So don't be afraid. Thank you so much.
Yeah, one. Kaga I'm also 12:30. I'm still more.
I don't know if Dr. Zay, have you dropped the question on the chat box?
That's exactly what I'm doing.
Please drop the question so that Dr. Famakide can answer. But Dr. Famakide, can you hear us? Can you?
I can't hear. I can't hear from here. I can't hear. Let's put something in the chat.
Sorry, Dr. Famakinde, can you switch off your video? Maybe that will help you. Switch off your video. Maybe your network is not strong to accommodate your video and audio at the same time. Switch off the video. Let's see whether you can hear. Click on the camera icon after the audio.
Hello? Okay.
Okay. Dr. Famakide, please go ahead. The question is on the chat box.
The message is incomplete. She said in terms of new drug molecule.
Hello, sir. Dr. Famakide.
Hello, you mentioned something about glucose uptake.
Yes,
Hello?
it's on the chat box. Yes, we can hear you. We can hear you. It's on the chat box.
Okay. The role of increased glucose level in proliferation of cancer in the system. Okay, as a natural product chemistry in trying to achieve apoptosis, at what point do we target with respect to new drug molecule? Yes, I mentioned something about glucose uptake. There is an issue of a glucose transporter which facilitates glucose transporter member SLC2A1. And this one is facilitates the transport of glucose across plasma membrane of mammalian cells. And this one will make the glucose available for the mesocyte cells. I think that's what you are talking about. And in this case, it will enhance after the stabilization, it will now enhance the multiplication of further cells, affected cells. And this leads to proliferation. That's what I discussed the other time, in which you have a GLUT transporter 1. And I said, Warburg effect, this is an increase in the rate of glucose uptake and preferential production of lactate, even in the presence of oxygen. So this will favor proliferation of the cancer cells. So then the second question that apoptosis, at what point do we target with respect to new molecule? You know, we have the cell circles. And we don't just administer chemotherapy for the fun of it. And each of them have their own targeted points. So for apoptosis, let me show you here so that you'll be able to understand properly. We have a programmed cell death. That is apoptosis. We have the normal cells that will go into cell shrinkage, whereby chromatin condensation occur, and there will be membrane bleeding. After bleeding, there will be collapse of there will be nuclear collapse. And this will lead to apoptotic body formation. So, I now said, if you really want to attack this, we have pro-apoptotic protein leading to the executioner phases. So if you want to do this, you will target the executioner phases in which the breakdown of a cytoskeleton will not occur. That's one. Then again, you can target it also from the initiator capsaicin. And once you do this, it will not further go down to the apoptotic body formation and also the formation of a cytoscomic bag. So all these points are the targeted areas. These are the targeted areas. You target the initiator or you target the executioner. So that is from the death receptor extrinsic pathway. But for intrinsic pathway, you can target the BCL2 family effector, which is BAS, and also the regulator here. So once you target this, or that one will not lead to pro-apoptotic protein, or you target from the initiator, which will not lead to executioner. So you either target from initiator or from regulator using the BCL2 or BCLX. So those are the target areas. So the regulator, then the initiator, the executioners. I hope you've gotten the question, my answers. Hello?
Thank you so much, Prof.
Hello?
Yes, I got you right. I got you right. Thank you. Hello, Prof. I got you right. Can you hear me?
Okay.
I got you right, sir.
Dr. Williams, is your hand raised again, or is it the former hand?
It's raised again. It's a comment, a write-on comment to his response. The information we are gathering as participants should actually make us better pharmacists. With this information with regards to the role of glucose in the initiation and progression of cancer, we want to always counsel our patients to go low on glucose consumption. When you have, and even for diabetic patients, hyperglycemia is a very strong driver. So we want to encourage them to go low. That brings to our mind public health initiative or programs that we can engage in so that we can all fight cancer and be victorious. Thank you, sir.
All right, thank you. Priska Egu, you have a question or a comment? Priska?
Priska, please unmute yourself and ask your question or make your submission.
Good afternoon, sir. Good afternoon, Dr. Telema. Can you hear me?
Yes, ma'am. Please go ahead with your question.
Okay, I just want to comment on what the last speaker said. I have raised this issue of glucose, as in patients not taking, trying to reduce their glucose level. But the consultant pointed out something to me, which I also think is correct. He told me that I shouldn't forget that there's something like gluconeogenesis. So even if the patient is not taking carbohydrates directly, that the body will still find a way of generating this glucose. So that's made, in fact, I'm actually confused on really, is it really correct to advise our patients not to be taking in, not to reduce their carbohydrate level? Thank you. Hello?
Thank you, Dr. Famakinde.
Put it in the chat
Did you get
box.
a question?
I think I couldn't have. Let's put it in the chat box.
Priska, Dr. Famakinde says, can you put it in the chat box?
Yes, that's what I'm trying to do. I'm trying to write.
Right. Thank you.
Okay. Hello, can you hear me?
Yes, we can. Go ahead.
I mentioned under the role of a microenvironment in tumor progression. And I specifically mentioned right from the opposite role. And here I said acidic pH suppresses immune cells and enhances invasion. So acidic pH in the microenvironment suppress immune cells and it enhances invasion, which leads to, which favors metastasis and tumor progression. Do you get me clear now? Hello? Hello?
Yes, sir. Yes, sir. We heard you.
Yes.
Please, there's another
Yes.
question, sir. Sir, there's another question in that chat box.
Okay. In our environment here in low resource countries, the issue of adequate nutrition is a problem. So we can we will not say our patients should not take carbohydrates, but they can reduce intake. And there's a difference between, we don't say balanced diet nowadays, but we say adequate diet, in which little proportion of the nutrients are available for our patients. So we will counsel our patients to reduce intake, but not that to the extent that to frighten them or to make them lose their hard-earned money that they are supposed to use for their chemotherapy or other treatment plan. So there's a way we counsel cancer patients, and we even take into consideration certain facts. It's a skill, because you too have to be versed in nutrition. So then you give a local available food, the mix for the patient. But we can say we can reduce a carbohydrate intake, but not to say it in such a way that the patient will now use that to jeopardize the family economy or at the detriment of the money available for their treatment plan. Thank you.
Dr. Thelma, over to you.
Okay.
Hello,
Thank
Dr.
you.
Thelma.
Yes,
Okay.
yes. Just a minute. Thank you very much for that presentation. For all the questions and the submissions that have been made.
We can't hear you again, no? Or is it only me?
I can't hear her either.
I'm sure it's network. Network is always a challenge. Hello, Dr. Thelma.
Hello, can you hear me now?
Yes, welcome back.
Yeah, I'm looking at the chart.
Okay.
Can it be safe to say or assume that stage 4 cancer patients can have their condition reversed to normal? If so, please help with possible therapy strategies. I want you to know that when we are formulating a therapeutic plan for cancer patients, we use certain criteria. Like you mentioned the stage, but the type of cancer matters. The type of cancer matters. So we cannot generalize that stage 4 cancer patients can have their condition reversed. We cannot generalize because some are more chronic, life-threatening than the other.
Dr. Famakide, can I contribute to that, please, sir?
I do.
I want to make a contribution to the question about reversing stage 4
you
cancer.
can. Because we have
Okay.
targeted therapy. We have targeted therapy if which are new molecules that are in vogue now. So you can contribute, please.
Thank you, sir.
The type of cancer matters, and also the stage, the location, and the location of the of the cancer cells. All those ones have implications in the outcome of cancer treatment. So please, more contribution.
Thank you so much, sir. I just want to say that when it comes to cancer treatment, there is always a goal. The goal of treatment is very important. For early detected early stage, the goal is always cure for stage 1, stage 2. But for locally advanced stage 3, stage 4, the goal is no longer cure. The goal is to delay disease progression, improve the quality of life. That's all. Cure is no longer there. But there is one remarkable thing we are noticing now. With the advent of innovator therapies, targeted therapies, antibody drug conjugates, checkpoint inhibitors, the immunotherapies, we are now seeing people with stage 4 cancers surviving and living longer life with the disease, but they will live with the disease, but they are very stable.
Adjusted life years.
Especially when they are on their treatment. You know, when you are stage 4, the treatment is lifelong. You have to be on the drugs. Till it's lifelong, till you are no longer responding to the drugs. Then we look at it and see if there is any other alternative for you. So we have HER2 positive stage 4 breast cancer patients doing fantastically well on antibody drug conjugate. In fact, there is one we have now that we reviewed yesterday. She's currently doing her chemo. This should be her 6th cycle today. Yesterday, when she came to my office for us to prepare her for today's chemo, I couldn't believe it. This was somebody that was on a wheelchair that could not move. She was on a wheelchair. She was so bad. By the time we started her on antibody drug conjugate, she had brain metastasis. She had bone metastasis to the lung, to the liver, everywhere. She's now working on her own. She's doing so well. The ulcers on the breast have healed. So what I'm trying to say is that you can do well as a stage 4 cancer patient if you can afford the current targeted therapies that we have. It's just that they are very expensive. But the good thing is that in Nigeria, most of these targeted therapies are available under NHIA. So please encourage your people to register under NHIA. I don't know about other countries, but in Nigeria, you can actually access antibody drug conjugate from Roche under the National Health Insurance scheme. And patients are doing well. Stage 4 cancer patients, they are stable, doing well on it. Thank you. They're not cured.
Thanks so much for that contribution. Quite enlightening.
In addition, like, thank you, my sister. You need to know that the goal of cancer treatment is not just cure. It's not just cure. She has mentioned it. We have quality adjusted life years, in which the patient will be healthy. You know, the outcome of care, clinical outcome, humanistic outcome, as well as economic outcome. So all those ones are also play important role in our caring for cancer patients. So we can extend the years that they will be available with us here, prolong their stay, arrest symptoms, and so on and so forth. So those are the things. The goal of cancer care, when it passes through stage, when it reaches stage 3, 4, then we have to look at other areas other than cure. Thank you.
Thank you so much, sir, and thank you, Fam, for the explanation. Yeah, because in most cases we see, we hear, not see now, we hear cases of persons say, oh, they were stage 4 and they got reversed and all that. So that's the reason why I asked that question, and all the rest. Now I know and more informed. So when a patient comes and says they were stage 4 and they have become normal, I now know how to advise them through farm care, provisions, and know what next to do. Thank you so much.
You're welcome. Dr. Thelma.
Yes. So I think from this time till about 12, we put there for Q&A. So we'll still take any more questions or submissions at this point. If there aren't any, then we'll take closing remarks. So in the next 2-3 minutes, if we're not getting any questions, contributions, or any submissions, then we'll take closing remarks, the closing prayer, and then end for today. Meanwhile, the participants, you are all supposed to complete the attendance Google form. If you have joined and you haven't done that, kindly go to the chat and do that. We'll repaste the link on the chat. So any more questions, contributions, submissions on today's topic or related?
Dr. Thelma, it's like you post the attendance link again.
The link, all right. Let me repost
Yes,
it.
please. Please, participants, please. what happened today, I don't know why it's difficult for most of us to obey instructions. Tomorrow, we will not allow any participant to join with their name. Prof pleaded today that we should just allow for today. Please, when you join, rename yourself to your participant ID. And if you've not received your participant ID, please post it on the group so that Dr. Selma can sort it out, please.
So I've reposted the attendance Google form on the link. So we'll allow a few minutes, 2 or 3 minutes for those of you who are yet to sign, to complete it, to do so. All right, so someone is asking, what is or are the differential tools for differentiating oncogenes from tumor suppressor genes? I'll take that again. Someone is asking, what is or are the differential tools for differentiating oncogenes from tumor suppressor genes? Thank you, Dr. Famakinde. Over to you. Sir, the question is in the chat box.
Okay. Let me check.
Dr. Famakinde, the question is in the chat
Hello.
box. Hello? Have you seen the question in the chat box?
Okay, okay.
Yes.
Let me just... Okay, differential tools for differentiating tumor suppressants.
Dr. Famakinde, we can't hear you. Hello, sir.
Okay.
Did you see the question on the chat box, sir?
Hello.
Did you see the question on the chat box?
Hello.
Go ahead and answer the question on the chat box, sir. Ask the question on the chat box.
Hello. Like I mentioned earlier on, I said tumor suppressor genes put a brake on the cell growth, preventing tumors from forming. But when they mutate, the cell growth continues unchecked. While oncogenes, in contrast, press gas cells growth and they multiply. So when you have a when you now have a oncogenes, we have oncogenes, mutation in one or two allele is sufficient to know that. But when you have tumor suppressant genes, both alleles will be affected. I have it on my slide 24, and I'm showing it now. Can you see it? Then mutation arises in somatic cells, not inherited, whereas mutations in the germ cells under tumor suppressant gene can be inherited. So then, the tumor suppressant gene often have strong tissue preference. So these ones can be seen in the tissues. Hello?
Hello, sir.
Can you hear me?
Yes.
Yeah, we can hear you.
Then we have a loss of functions of protein in tumor suppressant genes. Whereas in oncogenes, there's gain of function of a protein that signals cell division. So there will be cell division in oncogenes. And also under tumor suppressor genes, both alleles must be affected. But in oncogenes, mutation will only occur in one or two alleles, and that is sufficient to differentiate between tumor suppressants and oncogenes. Are you okay with that answer? Hello?
Serial number 016. You asked the question. Doc is asking if you're okay with the answer.
Hello?
Hello, sir. We can hear you. 016.
Okay, I'm okay. I'm okay with the answer.
All right. All right. Any additions to Doc's answers? Any questions? Please make sure you complete the attendance form. All right. I see there aren't any further questions or contributions. I'll just say one more time that we should complete the attendance form that has been put on the chat box.
Hello, Dr.
All right.
Thelma. Do we need to complete it twice? It's just once for
It's
today?
once. Please,
Okay.
it's once.
All right. Thank you.
All right. So I want to thank you all. I want to let us have at the back of our minds the outline for tomorrow, 14th March. We are starting at 9 a.m. prompt. And the topic, we'll first have a recap of today's session. So we need to have you, a number of you who joined today, give us a recap tomorrow morning, just after the opening prayers. And then the topics for tomorrow is cancer epidemiology and risk factors. So we'll look at the current landscape of cancer in Nigeria and globally, a burden of disease, common cancers like breast, prostate, and cervical cancers. Yes, there's a hand up. Kindly unmute and then, yes.
Hello,
Yes.
good afternoon. Thanks for the wonderful presentation. I cannot see the attendance on my chat box to register.
Please, it's there. Kindly check. It was just put there a few minutes ago by Prof. So go and
Okay.
check at the chat. It's there. And it's been put
So
again.
sorry, it's a link. If you just open the chat box now, you will see the link beginning with HTTPS, and all that. Click on it.
All right. Thank you.
And then paste it. Yes, it's there. I'll put it up again.
All right.
Please,
Okay. And
I
then
have a little challenge, please, before the closing prayer time.
please go ahead with your question.
Yeah, after answering my questions, the questions I submitted, then I now clicked view the score. I saw the score, but when I now went back to that fresh page, I noticed that my ID is showing red with X something something on it. So I don't know why it's like that, and I don't know if with that, if my the test will be recorded, or so I don't know the implication of that red that is showing there.
If you saw your score, it means it was recorded.
Okay, I saw my score.
It's recorded.
Okay, thank you.
All right. Any further questions or
Yeah, Dr. Thelma, I hope the material will be available to us today as we prepare for the recap tomorrow.
Yes, sir.
it will be
We'll make it
available?
available.
It will be made
Yes.
available.
It's all right.
All right. So, and then we'll end with social determinants of health and barriers to early diagnosis. The presenters tomorrow are Farm Vivian Osigale, Farm Omo Osi, and then Farm Oriyomi. We would wrap up around 12:30 to 1 with question and answer. And we'll take closing remarks as well. Prof, and then Madam Ijeoma, Prof Odile, over to you, sir, for your closing remarks.
Yeah, thank you, Thelma. Thank you for a wonderful job. Yeah, so we've come to the end of today's session. I want to apologize for the initial hitch we had with the presenter. Usually, when you're starting a big project like this, you have some teething problems that you don't really envisage. But thank God we overcame it. Tomorrow, let me just say, for the presenters tomorrow, please look for a place where you have free-flowing network and have at least 2 devices. I know right now we're in the rainy season in some places, so we have network challenges. Sometimes too, we have power failure. But please, let's try and get to a place where we can have seamless network so that we will not have the same issues we had today. And for our participants, not everybody who registered turned up today. I want you to know that you need to fill the attendance. For you to get a certificate, at least you should be exposed to the lectures. You should actually take the lectures and then do the pre-test and then the post-test. It's not just to pay and then want to get a certificate. We want, beyond all that, we want the knowledge to be passed on to our pharmacists. So I want to thank everybody today and hope to see you tomorrow. Please, let's come early. Tomorrow is Saturday. I know we do a lot of things on Saturdays, but let's come early. And tomorrow's lecture will not be as technical as today's, but that means also that we should get hold of the slides and try to digest. It will take some time to actually become familiar with all the different topics that Dr. Famakinde discussed today. So I wish us the best of today, the rest of the day. Thank you and see you tomorrow.
Thank you very much, Prof. And I agree with participants joining early and then going through the materials that will be made available today so that the recap tomorrow will be very interactive. Pharmacist Ijoma, your closing remarks.
Thank you so much. I want to appreciate everybody for making our workshop a success today, despite our at least we were able to okay. Hello, can you hear me?
We lost you after a while, but we can hear you now.
Okay. Okay, thank you so much. So like Prof said, Prof has said everything. Tomorrow we will resume by 9 a.m. and we'll go straight to the recap of today's presentation and then tomorrow's presentation. So please join on time and remember to change your name to participant ID so that there will be no need to remove everybody and no need for anybody to want to rejoin. If we just simply obey instructions, it's a simple instruction. Please, if you don't know how to rename your name to your participant ID, practice it now after today's presentation so that tomorrow it will be easier for you. The link for tomorrow's presentation will be shared. It's not this today's link that we are going to use. We are going to use a new link. So all the people that were not able to rejoin, they will be able to join us tomorrow. And the video recording for today's presentation will also be made available, as well as the slides for the presentation. And like I said, there's another online course where all these topics are well explained that will make available to all the participants at the end of this workshop. So you can, at your own convenient time, also do that online course. It will help you to understand oncology pharmacy better. Thank you so much.
All right. Thank you very much, Madam Ijoma. Can we get a closing prayer? Then we can officially end this session. Closing prayer. Serial number 022. 022. Please pray for us to end.
I said the opening prayer. Do you want me to also take the closing prayer?
It's fine. Please take it for us.
Okay. Our Father and our God, we are here again to say thank you for how far you have helped us today. We thank you for being with us all through today's training. We thank you for the facilitator. Thank you for the participant. Thank you for the administrators. We pray that you will continue to help us to get the best out of this training. And as we come again tomorrow, we pray that you will make it possible for us and even for more people to join in. Father, we commit the rest of the day into your hands. Be with us in all our various activities. And at the end of today, let us have every cause to glorify your name. For we pray with thanksgiving in Jesus' name. Amen.
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 Thank you so much to everybody.
Thank you so much.
Participants, Pan, Clinical Pharmacy Executives, Prof. Odile, Dr. Onodugu. Thank you to all of you and see you tomorrow morning.
Thank you all.
Thank you. Bye, everyone.
Have a lovely day ahead. Bye.
Thank you.